there are many new treatments coming to clinical trials in the next couple of years based on a really good mechanistic understanding of the disease. At multiple points in the cascade of cause and effect leading to these disorders, there are many, many targets. So I think one of the challenges is really to make that message known, to be really positive about the prospects for progress towards definitive treatments. I think it needs a concerted, multi-centre, multinational effort, which fortunately the MDS has been very effective in supporting through the PSP study group. We need to try, I think, at the first phase, that we trying these disease-modifying drugs in isolation, one at a time. But as in cancer therapy, as in HIV therapies, the real powerful therapies come from combinations of treatments. So already we need to start thinking ahead to combination treatments. Is it going to be a combination of anti-inflammatory, anti-tau, anti-UPR modification to really get a benefit for patients? So I think that'll be a few years down the line, but it's something we should already be thinking about as a community, how to organise and deliver such trials. The other thing I think which is very exciting is not just the advent of clinical trials, it's we now have, in the last 5 years, a much broader ways to understand what's happening in patients in the early stage while they're alive. So again, you might talk about these as biomarkers, but they're also mechanistic tools to quantify what's happening in humans with PSP and humans with corticobasal degeneration. Although there are genetic mouse models of genetic tauopathy, they don't really recapitulate many aspects of these illnesses. There are things that are uniquely human. And so to really be able to treat the human disease, you need to understand the human disease, which means you need to measure what's happening in our heads during the development of our tauopathy. So there are new PET ligands that have come online to enable us to do that, looking at inflammation and tau and synaptic loss. So particularly excited by this ability to measure synaptic loss is one of the earliest effects of the pathology. We can also use advances in MRI and the ultra-high field MRI to measure very detailed structures in the brain with tremendous sensitivity with molecular imaging from MRI, not just from PET. And the third is, I think, a new consensus on how do we share data, how do we collaborate in an open science framework to really move more quickly as a community towards the eventual treatments for our patients. And those treatments, I would say, need to think about symptoms as well as disease-modifying drugs. If we merely slow the disease down, in some sense, we've helped patients, but we've also created a bigger problem. We would have many more patients living with symptoms. So any effort to slow the disease down means we also have to deal effectively with the problems that our patients have, their symptoms. And there is, I think, good evidence now of new ways that we could try and tackle individual symptoms that matter. Apathy is a nice example, a symptom that matters to patients and families, and it affects prognosis and survival. Let's think about treating that as a symptom alongside trying to moderate the course of the tauopathy itself. And I think that's a very exciting prospect for combination treatments to slow the disease and to treat the disease, always focusing on the human disorder.